Perfect. Well, thanks so much for the opportunity to be here. My first time in Montreal, really enjoying it. So thank you for the beautiful weather. So my lab and I are really obsessed with all things protein dynamics and particularly their implications for drug discovery and precision medicine more broadly. And so what I'd like to do is introduce a couple of the features that we're really interested in that protein dynamics give rise to that have really interesting implications for drug discovery, and dive deeply into one of our favorite systems of late, and then show you how we're using machine learning to take this to a much larger and broader scale. So probably you're all familiar with looking at protein structure. So this is a surface representation of a class of enzymes called beta-lactamases that we've done a lot with in my lab in the past. And these are really great systems from a drug discovery perspective, because you can look at this surface representation of the structure and you can see this nice pocket here that is the active site of the enzyme. And you can say, great, this is a wonderful candidate for hunting for a small molecule that binds tightly to this pocket and prevents the enzyme from engaging its substrate so you can inhibit it. So with a lot of effort and a big helping of luck, you can use this structural information to help guide a drug discovery program and find such inhibitors. And this is great, but there's a couple of challenges. One is that it is hard, which is a little concerning that maybe we're missing something. And secondly, if you go and look at many other proteins like this transcription factor, you say, oh, well, now we have a problem because the structure is high quality, but there's no pockets. There's nowhere for a small molecule to bind with the high affinity that we would like for drug discovery by forming all of these interactions in every possible direction, basically. And so based on criteria like this, it's common to come to the conclusion that most of the proteome is not druggable, right? So the National Cancer Institute, NCI, on their website, explains this in layman's terms and comes to the conclusion that only 15% of proteins implicated in cancer are viable drug targets. And this is true across the board. And so this is a major challenge if you are thinking about things in terms of these individual snapshots that we're so used to looking at, and all of the rich information that's there. And while this is highly valuable, I have a very different intuition for proteins that I want to share with you through this video of a protein folding that we generated a while ago now. And so at the beginning, what you are going to see is this extended chain, the backbone of our protein. And we have a couple of the core hydrophobic residues that are the interior of the protein spheres, and some of the other side chains of amino acids are in sticks. And so you can see that as this progresses, we get collapse. As we talked about earlier, we start getting some of the secondary structure elements, so helice in purple, and yellow. And I'll be quiet now and let you watch the rest. So AlphaFold, which we've talked about extensively now, and OpenFold, they're all about predicting these experimentally observed structures, which again is like amazing and highly valuable. There's this other aspect to protein folding, which is how do you get there? How do you get from this extended chain? And this very highly dynamic process of folding or self-assembly also has broad implications, because as you can see, this doesn't just lock into the one static structure at the end. There continue to be fluctuations. And if we continued the simulation out longer, we would see the protein go so far as to spontaneously unfold and refold. And this creates some interesting opportunities. So, and explains some surprising observations from the literature. So just as an example, one study that really inspired my passion for this is a case where people were trying to use computational docking algorithms to hunt for inhibitors of these beta-lactamase enzymes that I've showed you. So they applied, the state-of-the-art docking tools at the time. They found some compounds that looked like they formed really nice interactions in the active site. So said, great, these are very promising. They went on to show that the compounds inhibited the enzymes experimentally. So that great, we've really got a success story here. So they went on to solve the co-crystal structure of their compounds bound to beta-lactamase. And now things got surprising because instead of finding this ligand-free or apo structure in blue with the compound nicely nestled in the active site, we got this yellow structure, right, with the compound in cyan over here, right? And so this was really puzzling because first of all, the compound doesn't bind where they expected it to. Secondly, the pocket where it binds is what we call a cryptic pocket because there's no evidence of a pocket in the ligand-free structure. One would presume a priori these helices are packed up against each other and there's no pocket there. And thirdly, something we won't spend much time on is not much has changed about the active site. So it's not obvious why these molecules are inhibitors. And I thought that was fascinating because it's popped up a number of times in the literature. From talking to people in pharma, there's many unpublished cases of this. And inevitably, when people discuss it, they fall victim to the temptation to say that this is an induced fit phenomena. So clearly, the protein must exist in this conformation. And so this ligand is somehow coming along and causing the protein to transition to this yellow structure. But my intuition seeing this was very different, was that if we could sit and watch the sorts of fluctuations that a protein undergoes at thermal equilibrium, we'd be able to see things like spontaneous separation of these helices to form this pocket. And then we could start using that information to inform the design of small molecules that would bind this cryptic pocket and potentially allosterically influence distant sites. And so we've done a lot with developing active learning algorithms, building off of molecular dynamic simulations. I won't go into that in great detail, but we'll focus on the machine learning we've been doing. But I want to show you, one of the systems we've been working on and some of the tools that we've come up along the way. And then again, how we're taking this to a broader set of proteins. So this particular example is a protein from Ebola virus called VP35. It performs a number of functions, this little 120 residue domain, but one of the best understood is binding to double-stranded RNA that is formed when the virus copies its genome. And our cells don't have much use for double-stranded DNA, and so we've got these innate immune mechanisms that are always on the lookout for the presence of double-stranded RNA as a telltale sign of a viral infection that will trigger an immune response. And so what this protein is doing is binding to the blunt ends and coating the backbone of this double-stranded RNA to prevent our immune proteins from binding to it and triggering this immune response. And so people have shown that this is absolutely essential for Ebola's replication cycle. If you make mutations that weaken this blunt end binding mode by as little as like 3 to 5-fold, in fact, our immune systems are great at recognizing an Ebola infection early on and terminating it, long before it develops into this, nasty, lethal hemorrhagic fever that gets the news media all riled up and makes us wonder if we want to travel, right? And so from this perspective, this VP35 protein is a great drug target, absolutely essential. You don't need to modulate this protein RNA binding affinity very much to be useful. But if you look at a surface representation of these binding interfaces, we come face to face with this problem of, oh, well, there's no nice deep pockets there. So what are we going to do? So we applied all of our active learning and simulation algorithms to this, and we found this beautiful cryptic pocket. So this is a morph, so it's smoothed out. The actual dynamics are far more jittery, but it gives you clearly the sense that there's this large flexing in the middle of the protein that creates this big pocket. That pocket is appealing in the sense that it's large enough to accommodate drug-sized molecules. There's plenty of interesting chemistry around, so you could imagine getting something that binds specifically. One of the other challenges, though, is that it doesn't directly coincide with the most important RNA binding interface, which is off to the left here. And so our next question was, okay, well, we have this pocket. Is it structurally or allosterically coupled to this key RNA binding interface such that targeting with a small molecule would do anything, right? And so to answer this, what we wanted to do was, do a comparison of, what the structure of this RNA binding interface looks like when this cryptic pocket is closed, as in all the crystal structures, versus when this pocket is open, to see if there's a nice delta. And this turns out to be a hard problem because we don't have, a single closed and a single open structure. We have ensembles or sets of open and closed structures that we want to compare. And in fact, this is a very general problem where, whatever we simulate, whether it's VP35 from Ebola or these beta-lactamase enzymes, we get these massive different structures. And it's very hard to, pull up thousands or millions of structures in your favorite viewer and say, aha, I understand. Here's how mutations are going to modulate function, and here's my favorite drug candidates. And so we spent a lot of time on thinking about how to analyze these large data sets and extract information from them. And coarse graining is a very important part of this. There's many algorithms for doing this. A favorite is PCA, or the time-lagged analog TICA. And this is great, but it can be problematic for these very complicated systems where it's not obvious what's important because they make various a priori assumptions about what is important. So for example, PCA and related methods assume essentially that larger geometric fluctuations are more important than smaller ones, which can be true, but isn't always true. Small fluctuations in the active site can make a big difference. And so if you apply PCA to two protein variants that you're interested in understanding, why is this variant more active than that one? Or why is this one more stable than that one? Or why does the open ensemble of structures differ from the closed? When you project this down into your coarse grained space, they overlap and it's hard to learn anything. And so we wanted to create a way of comparing ensembles that was more agnostic to these a priori assumptions and really just went after the biochemical property that you're interested in. And so to do this, we built off of autoencoders, which I imagine most of you are familiar with, but just to make sure we're on the same page, the idea is you take some high dimensional input, like Cartesian coordinates of a protein structure, you run it through a network that's our encoder that projects it down to lower and lower dimensionality till you get your latent space, which is your coarse grained representation of this data. Then you run it through a decoder that goes back up to high dimension, and you train this to minimize the difference between the input and output. And if you're successful, then your low dimensional latent space is a great coarse graining of your data. So this is a nice starting point because of the flexibility of neural networks. At the beginning, your standard autoencoder will have the same pathology as PCA, where it's intrinsically going to assume that larger geometric fluctuations are more important than smaller ones. But the nice thing is that I'm not a good enough mathematician to morph PCA to change that a priori assumption. But neural networks, you can start tacking things on. And so we developed a self-supervised autoencoder architecture where there's two key additions to your standard autoencoder. The first is that we ask the network to predict a label based just on the latent space. Right, and this can be very detailed in principle. So it could be that it really has to predict quantitatively differences in activities of different protein variants, 2 or more variants. It could be really simple. It could just be we're going to put label of 0 on low activity variants and label of 1 on high activity variants. And we're going to ask the network to be able to predict those labels based on the latent space. And so this is going to force the network to reallocate some of that representational power from large geometric fluctuations to whatever it is that's important for this label, even if it's a very subtle side chain swivel on the active site. So that's one of the key things. If you have thought a bunch about ensembles, you should say, well, that's nice, Greg, but you don't know these labels, right? You know that one variant is more active than another, for example, but that's an ensemble property. You can't tell me that hey, this structure is more active than that structure. And so the other key component of making this happen is that we use an expectation maximization algorithm. So we give things some initial labels. So we'll say, hey, everything from every structure from the low activity variants, we're going to give a label close to 0. Everything from the high activity will give a label close to 1. But now we're going to, update these labels after each epoch of training to converge on a self-consistent set of labels that recognizes that there's overlap between these ensembles. And you can't say, hey, the structure is high activity in this case and low in that case. It has to be, more or less the same activity. And so this has turned out to be really powerful. So now we can take these data sets that we've worked on comparing protein variants that alter stability. And when we couldn't distinguish them, based on coarse grainings with PCA or autoencoders, we can distinguish them very cleanly based on our DiffNats, these self-supervised autoencoders. And now we can bring it to bear, not just on comparing protein variants to make sense of their biochemical differences in terms of structure, but we can bring it to problems like our cryptic pockets. Before I get there, I was just going to show an example. So here's our initial labels. So we have in this case, we were looking at stability. So we assigned all of our low stability structures from our simulations to 0, high stability to 1. But then after the expectation maximization, you can see that this has relaxed and we're seeing significant overlap between these ensembles. And now we can go and look at exemplar structures with different labels and see what degrees of freedom are correlated with those labels to tease out what structural elements, trends, differentiate the high and low stability variants. So coming to beta-lactam V35, now we can compare structural ensembles where this cryptic pocket is open or closed. We find all kinds of differences, but one of the key ones is that there's this phenylalanine that swivels into the core of the protein when this pocket opens, or at least has a strong tendency to do so. And this was really interesting because that forms a key stacking interaction with the blunt ends of double-stranded RNA that we know from mutational studies is really important for that protein RNA affinity. And so what this tells us is that if we target this cryptic pocket with small molecules, we're likely to stabilize structures where this phenylalanine is flipped into the core and no longer available to engage with RNA, and to get a reduction in that protein nucleic acid affinity that could render Ebola susceptible to our innate immune systems. So at this point, roughly a third to half of my lab is experimental. And so, one of the exciting things is that we can take this from, our in silico model of reality and ask, well, how good of a reflection of the reality that we inhabit where Ebola kills people is this? And it works out very nicely. So we have a bunch of different experimental ways of testing, whether these pockets exist and are functionally useful. So we've got data showing that chemical labeling reagents can react with residues that you would think are buried based on the crystal structure, but get exposed to solvent when these cryptic pockets open. We can show that when we covalently jam things in this pocket so that it can't close, we actually inhibit this RNA binding affinity by about 10-fold, which is well beyond the threshold that we need for therapeutic relevance. But rather than go into those details, I'll just show you one piece of data, which is that given this insight, we, to the best of our knowledge, are the first ones to go develop a high throughput screen for this target. And just based on the insight from our in silico work that there is likely a cryptic pocket here, we now have about a half a dozen hits where when we scan across concentrations of our small molecule, in a real experiment, we see that we're knocking the protein off of RNA, and we have IC50 of about 1 micromolar, 1 to 3 micromolar, which is a perfectly legitimate starting point for a drug discovery campaign. And now we're getting NMR data and other structural data to back up that these ligands are binding where we expected them to based on our simulations. So this was a really nice point to get to with this one target. My lab has been active for about 9 years now. And over that time, we've done a lot to get to this point. But one of the questions in the back of my head has always been like, well, look, these molecular dynamics simulations aren't cheap. Now we can do things that used to take, 6 months on thousands of computers and a couple weeks on tens of computers, but that's still, a non-trivial investment of computer resources and a lot of waiting, right? So I don't want to scan across the PDB with something that takes 2 to 10 weeks per protein structure. And so what I've challenged my lab members with for years is like, how could we develop an algorithm that takes a single structure, like a crystal structure, like this one of beta-lactamase's input, and predicts if and where cryptic pockets are likely to form accurately enough that we could use this to triage potential collaborations or scan across all of the proteins in a signaling cascade of interest to figure out which are the best ones for us to go after from this perspective, or whatever you dream of by going to scale and looking across proteomes. It took a while. We tried a bunch of things that never saw the light of day outside of lab, but now we've got an interesting neural network deep learning approach. A couple of people have tried this in the past, and you might think like, oh, we have the PDB, this massive data set and all these advances in architectures and training algorithms. So this should be a great place to come at things from. But it turns out there's only order of 100 examples in the PDB of protein structures where there's an apo structure, ligand-free structure where cryptic pockets are closed, and a holo or ligand-bound structure where you've captured the open state. So it's really not that much data to work with that from that perspective. So one of the key things is that having worked on simulating proteins hunting for cryptic pockets and other interesting conformational changes for quite a while now, is we have a lot of data. So this is just a smattering of morphs and movies of trajectories from some of the systems that we've been working on. But you can see that there's quite a lot going on. We've worked on dozens of different systems. Our first paper on tackling the proteome of the SARS-CoV-2 virus and related viruses, we have 0.1 seconds of simulation, which is insane. We joke that it might be more simulation data than has been generated before in human history. And like, I'm not sure if it's a joke. It might actually be true. I don't know how to test that one, but it's a lot of training data, way more structures than we have available from the protein data bank. So what we decided to do is to use this as our training set and say, okay, we're going to train a neural network that takes a structure from a simulation as input and tries to predict the probability that each residue in that structure participates in a cryptic pocket in the next fixed time interval, say 50 nanoseconds is where we ended up. And so we did this. This is now available online through this tool called PocketMiner, but it's based on graph neural networks, particularly the GVP or graph vector perceptron algorithm from Ron Dror. The idea is that we take our protein, we build up a network representation of it. So our nodes are amino acid residues. They have scalar attributes like the charge and amino acid identity. They also have vector attributes like a unit vector pointing from the alpha to the beta carbon, for example. And the network is designed so that all the operations on vector inputs are done on the L2 norms of vectors. And so you get invariance to rotation and translation, and you mix that with your scalars. But the vectors never themselves, never get mixed with scalars and complicate things. So the idea is that our network takes again a structure as input, and what we're training it to do is, given that structure, predict the probability that over the next 50 nanoseconds, each residue will participate in a cryptic pocket. So at the end, you can think of getting this color-coded structure where we go from blue for low probability to red for high probability. And we can ask how well this does. And it does quite well on our training set or held out test set. So here's our ROC curve. We actually did this with both the graph neural network and a 3D CNN. They give pretty similar performance. It's quite good. We're doing way better than random. So that's good. This thing is learning something about simulations. And so now the next question is this transferable? Can we take what we've learned from these simulations and now start using them for our original objective of predicting if and where cryptic pockets are likely to form from say a crystal structure or a cryo-EM structure. And so we went back through the PDB and combed that for new structures and built up our own test set from recent structures that were never in any of our simulations, and this turns out to work quite nicely as well. So here we're comparing our PocketMiner algorithm to a previous published algorithm called CryptoSite that's based on random forest. You can see they give pretty similar performance. But the big distinguisher is that CryptoSite needed molecular dynamics simulations as input. So for a typical, 100 residue protein, it would take like a day to run this algorithm, which still adds up if you're trying to think about scanning across, proteomes. Our algorithm takes like, order of a millisecond. And so we can really, take this to scale and start looking at large numbers of structures and, giving computational or even more importantly, medicinal chemists, the instantaneous feedback that they so desperately desire from their computational workflows. And this works really well in tests. Here's the application of crystal structures. So one of the things that we did is apply this to a set of about 10,000 proteins that are implicated in cancer. A priori, in this given data set, people had said that, based on things like the available structural data and precedents in the literature for targeting these proteins, that about half of them were druggable and the other about half were undruggable. With PocketMiner, we predict actually that of the undruggable, quote unquote, proteins, more than half of those have cryptic pockets that make them viable drug targets. And 50 nanoseconds isn't a lot of time. That's less time than the air quotes instantaneous freezing time of these plunge freezing apparatus that the cryo-EM folks use to prepare their samples for cryo-EM and tell us they did it instantaneously. And quite a bit is happening. And so I think as we work, get more data and can take this out to longer timescales, that it will turn out even more of these proteins have cryptic pockets that make them potentially viable targets. So to test this, we picked out a couple of exemplars. So one example is a kinase called PIM2 that's of interest in cancer. We applied PocketMiner and predicted that there's a number of residues up near the top here that have a high probability of participating in cryptic pockets and are nicely co-located with each other. So based on this, you can imagine some cryptic pocket opening there if we could watch this protein's dynamics. And now we can say, okay, with our simulation tools, we can watch this protein's dynamics. And so this structure here shows an open structure, and the cyan kind of blob there is a space filling representation of the void that's available for a small molecule when this cryptic pocket opens. And I've shown you with the VP35 example, and we have a couple of others. We've been pretty successful in experimentally confirming these sorts of predictions. So this is very encouraging. One question which comes up frequently, and especially given the order of talks here and the focus on OpenFold and AlphaFold at the beginning, is can AlphaFold help with this, right? And I won't go into great detail, but the answer is kind of, some of the times at least. So we've taken this trick that others have used of subsampling the multiple sequence alignment and making predictions based on different subsamplings of that sequence alignment to get some structural heterogeneity. And we've done this for some of these proteins that we have crystal structures of ligand-free and ligand-bound structures, capturing open and closed cryptic pockets. And so here's a set of proteins we've dug into, and what we're showing is the distribution of RMSDs of the structures that we have, this ensemble of structures we generate with AlphaFold to the experimentally determined ligand-bound open structure of the pocket. You can see that for some of them, we do quite well and really nail it. Others we don't do so well. But one nice thing is that all of our simulation algorithms benefit a lot from having heterogeneity in the starting structures. And so what we've showed is that even for these cases where AlphaFold doesn't give you the cryptic pocket of interest, starting our simulation workflow from this set of AlphaFold structures gets to that pocket way more quickly than you ever would based on just starting simulations from the ligand-free crystallographic structure. And so, AlphaFold still has a nice place to play in all of this as we look towards hunting for more of these and trying to exploit them with small molecule drugs. So hopefully that gives you a sense of the myriad ways that machine learning is, helping us out. In addition, you are, more physics-based and experimental workflows. So everything from making sense of these complicated ensembles to telling us where to deploy our molecular dynamics based algorithms, to helping jumpstart the whole process. With that, I'll thank, my lab who's done the work, all the different people that have supported, our work over the last many years, especially as we dove in on COVID using this folding at home distributed computing project that I run that lets us generate these massive data sets that we're working with. And thanks again for the invitation to talk and for being here to listen.
Thanks a lot for the great talk. It's really interesting to see how we can really try to find cryptic pockets to enable like new kind of drug discovery that would not like just go on the traditional pocket, but really tackle undruggable diseases, undruggable targets. And thanks a lot for the great talk. And now we'll take questions at the microphone here.
Hi, Greg, great talk. So I wanted to ask you, so you mentioned at the beginning, right, the undruggable proteome, like proteins like transcription factors, right? And so I know I just showed a data point at the end on the percentage of undruggables that may have these cryptic binding pockets. Yeah. But a lot of these undruggable proteins have intrinsically disordered domains, which make them very difficult for any MD or structure prediction method to do well on them. What percentage of these cryptic binding pockets would you expect to be inside of those intrinsically disordered domains, and how would you be able to identify those?
Yeah, that's a really good question. So we've really focused on more structured domains so far. So we in this example with 10,000 proteins and whatnot, we actually just pulled out the well-structured domains and focused on that. The intrinsically disordered proteins are a really interesting extreme of this distribution of varying degrees of conformational heterogeneity that we see. What I can say for now, oops, let me go back to this slide, is that one nice thing is that all our simulation algorithms are very nicely transferable to this to disordered proteins. So this ApoE protein down here is very interesting because it's the strongest genetic risk factor for Alzheimer's disease. And despite the fact that we've known that for 30 years, very little is known about what the point mutations that modulate Alzheimer's risk in this protein are doing, because the latter half of the protein has extensive disorder and it's a real pain to work with experimentally. So we've teamed up with a single molecule spectroscopist, and we see like beautiful agreement between things like predicted single molecule Fret distributions and experiments. And so that's a nice place to be in terms of being able to start thinking about other things like cryptic pockets. And I'm reasonably optimistic. From our survey of structures of cryptic pockets, one of the interesting things that we found is that lots of the structurally characterized cryptic pockets are what we in lab call reverse pockets, where rather than part of the structure opening up, which is what we focused on at the beginning, you have things like domains hinging towards each other, and now you have a nice groove where small molecules combined. And so I think you could see a similar thing with disordered proteins, where there's reasonably probable co-locations of residues that can wrap around a small molecule locally. So anyways, long answer to I don't know for sure, but I'm hopeful. I think it's very interesting.
I enjoy your talk. Really enjoyed it. So I have a question about using MD simulations for training, for different proteins. So this characteristic times and time scale of motions for different proteins, opening and closing, is different. So how do you know when to stop MD simulation?
Yeah. No, that's a great point. We never really know a priori when to stop. And so, the answer ends up being subjective, as we stop if we've seen something interesting and then go do an experiment and find out that we have a bona fide prediction that we want to follow up on. This does become particularly interesting in things like PocketMiner, since we do limit it to predicting where the probability that residues will participate in a cryptic pocket in a fixed time interval. So I think there's a lot of value in extending that time interval, or asking questions like, should one vary what that is? But I don't have clear answers for now, but just the existence proof that doing this sort of thing can be quite useful.
Is the speed the main issue right now with the EMD simulations, or the accuracy is a problem as well?
The speed versus accuracy.
Yeah.
So all of the simulation data that was used in this work was generated with Amber 03, so the Amber force field from 2003, right? And so to me, this is a testament to the fact that I think there's plenty of room to improve force fields. I'm not going to claim that they're perfect, but in the comparisons we've done, most the force fields give very similar behavior at a qualitative level and are quite predictive. And I think this speaks to the robustness of lots of biological systems. So I think to be useful, we don't need the perfect force field, and that in many, if not most cases, sampling, comes down to speed is the main bottleneck for making useful predictions. That's for proteins. I'll say that for small molecules, the diversity of chemical space is far larger, and that's an area that would benefit from continued force field work more immediately.
Thank you. And if I can have a follow-up on that,
Yeah.
Apart from what you mentioned with AlphaFold, have you experienced with any enhanced sampling methods?
Oh yeah,
Do they help?
so we do a lot with adaptive sampling algorithms, where we're iterating between running simulations, building a map of conformational space, notably a Markov state model or MSM, and then using that map to decide where to run the next batch of simulations, incorporating both geometric criteria about what sort of structures we're interested in, statistical criteria for model quality. We've also done things like using metadynamics or replica exchange to get some broad sampling, and then to jumpstart our Markov model construction from constant temperature, constant pressure simulation. So yes, there's a lot of benefit for plugging in all kinds of different ways of generating conformational heterogeneity starting points for the simulation methods, and then following up with extensive simulations to get accurate and statistically well-resolved models of relevant distributions.
Thank you.